Okay, so these notes are notes on nerve cells. We've already defined the neuron as a nerve cell. So a neuron is a nerve cell. A nerve impulse is a change in an electrical potential. It is also known as a nerve signal, the message, action potential, and I've got action potential highlighted because it's important that you know that. So nerve impulse is also called a nerve signal. It's also called the message. It's also called an action potential. A nerve impulse is a change in electrical potential. A dendrite is a structure on the neuron that carries nerve impulses into the soma. It makes connections to other neurons and receives communication from other cells. So the dendrite carries the nerve impulse into the soma. It makes connections to other neurons, and it receives communication from other cells. The axon is also known as, it's also called the nerve fiber. It carries nerve impulses away from the soma. The axon carries nerve impulses away from the soma, and it's also called the nerve fiber. The axon terminal are button-like ending at the end of the axon. It releases neurotransmitters into the synapse. So it changes the signal from an electrical signal to a chemical signal. So as this signal is going through the neuron, it's an electrical signal. When we get to the axon terminal, then we have chemicals that are released, and so that's why it's changing the signal from an electrical impulse to a chemical signal. So the axon terminal is button-like ending of the axon, releases neurotransmitters into the synapse. So it is where the signal is changed from an electrical signal to a chemical signal. A synapse, a lot of times that, I don't have that on here. Okay, so a lot of times when you hear synapse, you hear that that's the gap between neurons. But probably a little bit more accurate is that's the junction between two neurons. So that's where they come together. So it's a region. The synaptic cleft, that is the actual gap between the neurons. So synapse is the junction, the place where neurons come together, and it's a region. This is the place where the dendrite of one neuron and the axon of another one are at. The synaptic cleft is the actual gap. Myelin sheath. The myelin sheath is insulation of an axon. It allows for quick transmission of nerve impulses. The myelin sheath is insulation of an axon. It allows for quick transmission of nerve impulses. Schwann cells. Schwann cells are cells that produce the myelin sheath. Schwann cells are the cells that produce the myelin sheath. Nodes of Ranvier. A node of Ranvier is a gap in the myelin on axons that allow the action potential to remain strong. The ion channels are located here, and the node of Ranvier lets ions in for action potential. So node of Ranvier is a gap in the myelin on axons that allow action potential to remain strong. Ion channels located here. Node lets ions in for action potential. And then glial cells. Glial cells provide support and protection for neurons. Glial cells provide support and protection for neurons. All right, next. So on my, left-hand side page, talking about different types of neurons. So this first one is unipolar. Unipolar neuron has only one nerve process extending from the cell body. Unipolar neurons are not present in humans. They're found in invertebrates, so things without a vertebrae. So unipolar neurons only have one nerve process extending from the cell body. They're not present in humans, they're found in invertebrates. Bipolar neurons, they have two attachments to the soma. So we have the dendrite and an axon. And so the nerve impulse moves from the dendrite to the soma to the axon. Sensory neurons can be bipolar. Multipolar. Multipolar neurons have many points of attachment to the soma. The most common form of motor neurons and interneurons are multipolar. So we have tons of attachments of dendrites, and then we have this attachment of the axon. The direction of the nerve impulse is from the dendrites to the soma to the axon. Multipolar is the most common form of a neuron for motor neurons and interneurons. And then we have the pseudopolar. Oh, I said it wrong. Pseudo-unipolar. Pseudo-unipolar. Only one extension from the cell body. So this is the soma, the cell body. I have one extension, dendrite, axon. The nerve impulse is still moving from dendrite to axon, from dendrite to cell body to axon. And this is another form that sensory neurons can take. Pseudo-unipolar. Next page, my diagram, we've got some specifics for our neurons. Let me move this up a little bit so we can get this all in at the same time. All right, so interneurons, efferent neurons, afferent neurons. We'll go over that underneath in just a minute. Interneurons are usually central nervous system, so brain and spinal cord. Efferent neurons are motor neurons. Afferent neurons are sensory neurons. So interneurons, here's the cell body. We have dendrites that bring the message into the cell body. Nerve impulse moves along the axon to the axon terminals at the end of the axon. Notice that the efferent and afferent neurons are set up a little bit differently. They have the myelin sheath, which are these little bubbles on here. Interneurons, a lot of times they don't need that myelin sheath because they're packed so closely together that they don't need insulation. Efferent and afferent neurons might have to go from the brain all the way down to the tip of your toe or out to your arm, so they need that insulation in order to keep that message protected. So efferent neurons, here's the dendrites taking the information into the soma to the cell body. Then the impulse is going to move along the axon. So these little bubbles here represent the myelin sheath, and the gap in between them, those would represent the nodes of Ranvier. So this is a node of Ranvier, node of Ranvier, node of Ranvier, node, 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 node. At the end of the effector neuron is an effector organ. So it could be a muscle or a gland, something that's going to do something that's going to have an effect. So here's all my axon terminals. That's going to end at an effector organ. So this looks like a muscle fiber. So the signal would come down here. That signal would go across that synapse, would go across that gap by a neurotransmitter. The neurotransmitter would then cause a release of calcium in the sarcoplasmic reticulum, which then would result in a contraction. Afferent neurons. So afferent neurons, these are receptor cells. So it might be photoreceptor, chemoreceptor, something that's going to pick up information from the environment. So photoreceptor, like rods and cones in your eyes, they pick up different amounts of light or colors of light. Then the dendrites that are right here, that information gets passed along those dendrites. Notice that we have myelin sheath, these little bubbles, myelin sheath. We have nodes of Ranvier, node, 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 where the gap is. The message is going to move down to the cell body, down to the axon, and then to the axon terminals, which this would end up in the central nervous system. This one would start in the central nervous system and end up at the effector organ. All right, so sensory neuron. is also known as the afferent neuron. Sensory neurons pick up sensory information from the inside/outside of the body from receptor cells. So you have to have the specialized receptor cells to pick up the information. Sensory neurons themselves, they only carry it from your receptor cells to your brain. They don't pick up the information from the environment. Motor neurons, they just carry the message. They carry the message from the brain to the effector organ. So motor neurons are also called effector neurons. They send a signal, carry a signal to a muscle or gland, which would be the effector organ, that responds to the nerve impulse. So for example, the muscle fiber contracts, or if it's a gland, the response would be the hormones released. Interneurons are also known as association neurons. Interneurons are going to interpret/process sensory information and determine how to respond. So these guys are in the brain and spinal cord. So when the afferent neuron brings that information that was picked up from the receptor cells, the interneurons decide what to do with it and then give the message to the motor neuron, which takes it to the effector organ. Finally, you'll have a nice pretty color picture, but this is showing fluorescence in neurons on the picture. All right, so fluorescence. This is when an object/cell/substance absorbs light or energy and then emits visible light of a longer wavelength. So the object, the cell, the substance, is able to absorb energy, but it's not happy with it. It can't hang on to it. So it has to give it off, and when it gives off that light/energy, it emits it in a longer wavelength of light. So it's usually something that we can then see. A lot of times this is some kind of light or energy that we can't perceive, we can't see it, but the fluorescence is something that we can see, or we might be able to pick it up with some sort of diagnostic tool. Fluorescent microscopy, there we go. That's an imaging technique that's based on fluorescence. So they use this process to their advantage. So it's an imaging technique that's based on fluorescence. Researchers can target specific tissues and organelles with different colors of chemical markers to make them visible. So if they want to see where cells are going, what cells are doing, what's happening here or there, they can use fluorescent microscopy to tag cells with something that is going to fluoresce, something that's going to glow, so that they're able to see it and see what's going on.